Hi, I'm Jo. So I'm just going to give you a little brief talk through today on prokaryotic cells and basically the structure of them. So we're going to focus on in this video is part of the AQA A-level specification, which is 3.2.1.2, which is structure of prokaryotic cells and viruses. So to begin with, what I want to really do is go back to GCSE and let's have a little look at that original structure that we have been looking at previously. So at GCSE, one of the things you may have looked at is bacterial cell structures, prokaryotic cells as an example. And this is an image taken from the GCSE revision website. So what we're looking at here, the structures at GCSE you were expected to know about, and the little bits of detail is that a bacterial cell would have a cell membrane and that it'd be surrounded by a cell wall, and it would have a cytoplasm which contained ribosomes but no chloroplasts or mitochondria. It would also have included the idea that DNA is there as a single molecule and it's found free in the cytoplasm. And they also, in many cases of GCSE specifications, would have included the fact that DNA can also be found in small loops called plasmids, but that may not have been covered on your spec as much. You may have covered a little bit more about these small loops of DNA that you can find in bacteria if you also studied a little bit about genetic engineering. So you might have had both of those bits. So what we're looking at A-level is we have a little bit of a step up. So a little bit of a step up in the details surrounding the structures of the prokaryotic cells that we're expected to really get to grips with and understand. So let's start with the first bit. The first and most important thing is understanding your definition of a prokaryotic cell. So prokaryotic cell at A-level, we always talk about having no true nucleus or membrane-bound organelles. So it touched on this a little bit at GCSE, but what we're really focusing on here is that idea, no true nucleus or membrane-bound organelles. We've got a little diagram on the left that sort of starts to show some of the structures similar to what we'll have seen at GCSE, but we're adding that little tiny bit of detail to it now. So let's start with the first bit, the DNA. When we're looking at a prokaryotic cell, we will have a circular piece of DNA. It is not contained within a nucleus. Unique feature of all prokaryotic cells or bacteria. The DNA is not contained in a nucleus. It's circular, and it's in a loop form, and it's found free in the cytoplasm. It is not associated with proteins. So it's not wrapped around histones or packing proteins. It is free. It is, and again, that circular form. It is not linear as you would expect to see in a eukaryotic cell. The circular DNA and the position it's found within the cell sometimes gets referred to as the nucleoid, and that means it's not a nucleus, a nucleoid. The nucleoid is just the area within the cytoplasm that the DNA is found. Occasionally on textbooks you might see the DNA being referred to as the bacterial or prokaryotic genome. Now, the biggest, chunkiest circular piece of DNA that we find within our prokaryotic cells, this is where we find the genetic information that's needed for the replication of the bacterial cell. This is the essential genome that allows the survival of the cell. So it's essential genes, all the things that will be required to produce the enzymes required for genetic replication and binary fission and replication of a bacterial cell. So it's a really, really important set of structure within that circular DNA. Surrounding the cell, surrounding the cytoplasm, we refer to a cell membrane at GCSE. We're talking about this plasma membrane, and the plasma membrane simply controls the entry and exit of molecules into the cell. It decides what can come in and what can leave, and gives that, it's not just about allowing, it's a controlling measure. So it's a really, really important feature of our prokaryotic cell. Surrounding the plasma membrane is a cell wall. Now, you would have at GCSE just referred to this as a cell wall. Now, at A-level, with that added level of detail, what we need to know is, do we need to be able to distinguish the cell wall of a prokaryotic cell from a different cell, such as the eukaryotics plant cell or a eukaryotic fungi cell. Now, what is the distinguishing feature? They've all got a cell wall, but what we have that's different here is that the prokaryotic cell is made of a different component. Its prokaryotic cell wall is made of murine, which is a glycoprotein, so a combination of carbohydrate and peptides. Different prokaryotic cells have slightly different structures, but the murine being a central part to it. In some textbooks, you might see the cell wall being referred to as being a peptid or glycan cell wall. So it's just saying that it's a glycoprotein. But it's so important that when we're talking about the cell walls of prokaryotic cells, that we don't just refer to it as cell wall. We add that little bit of detail in. We put the murine label in as well. Now, surrounding that cell wall, we can also have what we call a capsule. Now, the capsule surrounds the complete part of the cell, and it protects the bacterium both from other cells, but also from dehydration if it was found on a surface and exposed for large proportions of time. So it's a protective layer. It's sometimes in some textbooks referred to as a slime layer because it's a slimy capsule. It's the chemicals that make it up give us that kind of texture or sort of properties to it. But again, a really important feature of our prokaryotic cell. Now, what you might notice as a little tip for A-level here is as we're labelling up this diagram, we're adding the labels up with label lines that join at 90 degrees and with a straight line. No arrowheads, touching the feature that we are identifying. And this is important because if you are expected to be able to label diagrams effectively as part of one of the A-level skills. And with that, these are the things that we're looking for: 90-degree angles, draw your label lines with the ruler. We are not wanting arrowheads, and just touching the feature that we are aiming to label. So we've looked at the sort of the structures that make sort of the outer layers. Let's go back into the prokaryotic cell and look at some of the features that we can find within it. One of the ones from GCSE that was mentioned is that the cytoplasm does contain ribosomes, and it does. It contains 70 S ribosomes. Ribosomes are an organelle, but they are not membrane bound. They are made of protein and ribosomal RNA. They do not have a membrane. They're made of two subunits, a large and a small subunit. But actually, the ribosomes that we find in prokaryotic cells are small. They have 70 S. They're smaller than the ribosomes you'd find in your eukaryotic cell. The ribosomes, like we've learnt previously, is the site of protein synthesis. Specifically, it's where translation is going to occur, and they're found free in the cytoplasm. The cytoplasm, exactly the same as we've seen before. The key thing here, prokaryotic cells does not contain any membrane-bound organelles. So I know at this point you probably think I'm sounding a little bit like a broken record, but it's a key important bit of information we've got to keep reminding ourselves. No membrane-bound organelles. So it's not going to have a mitochondria. It is not going to have chloroplast. It's not going to have a rough endoplasmic reticulum or a smooth endoplasmic reticulum or a Golgi apparatus. All of those organelles have got their own membranes and therefore will not be present in a prokaryotic cell. It will be the site of many chemical reactions, though, that occur within the prokaryotic cell. I'm just about touching this thing, so you can see the smaller circular pieces of around here. These are actually plasmids. You may have just heard of them as small circular loops of DNA at GCSE. You may have been given the term plasmid, but what we're looking for A-level is this term plasmid. So a plasmid is a circular piece of DNA, and it's important to recognise that it is smaller than the main circular DNA that we find, or this we refer to as the bacterial genome. Plasmids have a really important and quite a unique structure and feature to them. The plasmid possesses genes that may aid the survival of a bacteria in adverse conditions. It's not guaranteed, but they can have beneficial but not essential genes. So the main circular DNA has the essential genes that allow the bacteria to carry out its main functions and to replicate. The plasmid DNA will have beneficial ones. So genes that may allow it to survive despite the use of an antibiotic. So antibiotic resistant genes that will produce enzymes that mean that an antibiotic is not effective, or proteins that mean that an antibiotic's not effective. So it's really, really quite important. Plasmids can be replicated and swapped between bacterial cells. If they're in adverse conditions and they're stressed, they will swap them and obtain them from the environment as well, in the hope of increasing the potential of survival. So the last little main feature that we've got on this diagram is that of the flagellum. So the flagellum is is basically used for movement. It can have a whip-like motion, and it can help propel the prokaryotic cell through a medium. Some bacteria have many, some have few, some have none. It's very different. E. coli, as an example, can have many, many flagella. So then when we talk about numerous flagellum, we talk about flagella as the plural of it. So they can be numerous in number, they could be singular. And they're like projections that come out through the cell wall, through the capsule layer, and they can whip them around in a movement to actually propel a prokaryotic cell forward. Now, key things to remember about our prokaryotic cells is that they are smaller than eukaryotic cells. They're usually between 0.1 and 10 micrometers in length, so they're very, very tiny. Back to that key feature again: no nucleus or membrane-bound organelles, and a cell wall that is made of murine. So those key little things that we want to talk about. The bottom right here, I have got an electron micrograph image of a bacterial cell, so a prokaryotic cell, and we can see that we have got the nucleoid region where the circular DNA, the main DNA, is going to be found. You can see there's plenty of ribosomes present within that cytoplasm, but no membrane-bound organelles. We can see that we've got this large cell wall structure on the outside of that cell as well. So some of the main features that we've got, we can identify on our prokaryotic cells. There are a couple of prokaryotic cells, I say these are slightly beyond the specification, but they're useful to mention and discuss. One of them is the pili, or pilus as a singular. We can see it on this diagram. Now, they are different to the flagella. The flagella will go and connect with the plasma membrane directly, and have a connection through to the cytoplasm to allow for that whip-like movement. Pili are not going to connect in exactly the same way. Now, pili, or pilus as the singular, what they're there for is to facilitate cell adhesion. So they allow bacterial cells or prokaryotic cells to adhere to one another, to clump together. So again, quite a useful little feature for them. The other little thing that I wanted to really include beyond the spec is a little feature that's up here on this diagram, and it's an infolding in the plasma membrane. It's called the mesosome. It's where the plasma membrane folds up many times, so it increases the surface area. Now, the mesosome has multiple functions that we can just that can have been linked to it, one of which is a site of respiration. So it increases the area, surface area of the plasma membrane, allowing it for a good site of respiration, but it's also, we believe, involved in sort of secretion processes as well. So there's quite a few little functions linked to the mesosome. And to sort of finish off, I just wanted to introduce, so this is a sort of slightly computer enhanced image of an E. coli. So you can see here the E. coli structure, where you've got these massive whip-like flagellum coming off it to allow for that propulsion, that movement of the E. coli through the media, to allow it to get to different sources and move around. So it's just nice to see some sort of a bacterium in action from where we've been looking at before. So E. coli, notice at the top, is also Escherichia coli, is its official sort of binomial name. Notice it's written in italics with the coli portion in lowercase as the species name. So its species name is always in lowercase. So it's a little bits of tips and things that are useful to identify and name different prokaryotes and bacteria. I hope you found this a useful introduction into the structures of prokaryotic cells, and thank you for listening.